Bismillahirrahmanirrahim. Welcome again to Derma Immunology. I am Naglaa Elmongy, Professor of Dermatology, Cairo University. Our video today will be about transcriptomics in acne pathogenesis and the impact of isotretinoin treatment. Here is my reference in this video: Acne Transcriptomics, Fundamentals of Acne Pathogenesis and Isotretinoin Treatment, which was published in Cells in November 2023. Transcriptomics is the study of the transcriptome, which is the entire collection of RNA transcripts generated by the genome in specific conditions or within particular cell type. By comparing transcriptomes, it is possible to identify genes that show varied levels of expression in different cell populations or under various diseases or treatment conditions. Significant scientific advancements have recently been achieved in understanding the transcriptional regulation involved in acne vulgaris. Acne vulgaris, a prevalent chronic inflammatory skin condition, has a complicated mechanism of development. Four factors play vital roles in the pathophysiology of acne. Number one, the increase in altered sebum production, hyperseborrhea and dysseborrhea. Number two is the altered keratinization of the pilosebaceous duct or comedogenesis. Number three, effects mediated by Propionibacterium acnes. Number four, the T helper 17 cell driven inflammation. The onset of acne is triggered by multiple growth factors, which channel their signaling pathways into activating the kinase AKT ACT or protein kinase B. This is their main way. The activation of this kinase in turn modifies the function and expression of key transcription factors that play a significant role in exacerbating the condition. So during puberty, the signaling of insulin-like growth factor 1, insulin and androgen leads to the activation of the kinase ACT and the activation of mechanistic target of rapamycin complex 1 or mTORC1. Also, Western diet characterized by high glycemic carbohydrates and milk dairy products further stimulates the ACT mTORC1 signaling pathway. The mechanistic target of rapamycin complex 1 or mTORC1 is a key regulatory kinase that is responsive to growth factors and nutrients, playing a vital role in controlling cell proliferation and anabolic processes and suppressing autophagy. Activation of ACT will lead to phosphorylation of the nuclear FOXO1, which is the abbreviation of fork-head box protein 1, and FOXO3, which is the abbreviation of fork-head box protein 3, resulting in their relocation from the nucleus to the cytoplasm and preventing their inhibitory action on transcription factors involved in lipid production and inflammation. So this process will lead to the activation of several transcription factors, including the androgen receptors, steroregulatory element binding transcription factor 1, peroxisome proliferator activator receptor gamma, and signal transducer and activator of transcription 3, STAT3. On the other side, this shift results in a decrease in the FOXO1 dependent expression of GATA binding protein 6, GATA6, which is the key transcription factor responsible for maintaining the homeostasis of infundibular keratinocytes. Another important effect from activation of ACT is the phosphorylation of the p53 binding protein MDM2, which will lead to accelerated degradation of the p53 protein. Now I'm going to talk about insulin-like growth factor 1. Insulin-like growth factor 1 acts as a powerful mitogen binding specifically to the insulin-like growth factor 1 receptor located on the surface of targeted tissues. In the period of puberty, the enhanced production of growth hormone activates the growth hormone insulin-like growth factor 1 axis. Insulin-like growth factor 1, a critical hormone during puberty, facilitates sexual differentiation and the linear growth of bones. Acne typically appears at a later stage in puberty, coinciding with or following the peak of height growth in boys, and onset of menarche in girls. Also, the consumption of Western diet, particularly high in hyperglycemic carbohydrates and milk, has been linked to the development of acne through insulin-like growth factor 1 mediated pathways. Individuals with Laron syndrome, a syndrome characterized by congenital deficiency of insulin-like growth factor 1, are short in stature and do not develop acne unless they receive treatment with recombinant insulin-like growth factor 1. Upon insulin-like growth factor 1 binding to its receptor, the activation of phosphoinositol 3 kinase or PI3K occurs, leading to the subsequent activation of protein kinase B or ACT, as I said before, a key regulator in growth network control, and among its various regulatory effects, acts, phosphorylates, and thus inactivates the transcription factors FOXO1 and FOXO3, which are believed to have a significant role in the development of acne. And as I explained before, following its phosphorylation by ACT, FOXO1 is relocated from the nucleus into the cytoplasm, and in the nucleus, FOXO1 functions as a repressive co-regulator of the lipogenic transcription factors such as androgen receptors, steroregulatory element binding transcription factor 1, and peroxisome proliferator activator receptor gamma, as well as the pro-inflammatory signal transducer and activator of transcription 3, STAT3. FOXO1 interacts with STAT3, obstructing STAT3 ability to engage with what's called SP1-POMC promoter complex. This interaction consequently prevents the leptin-mediated actions of STAT3. Leptin is known to encourage a pro-inflammatory lipid profile and activate inflammatory pathways. So, enhanced insulin-like growth factor 1 signaling through the ACT-mediated inhibition of FOXO1 amplifies the transcriptional activity of androgen receptor, stereo response element binding transcription factor 1, PPAR gamma, and STAT3. These key transcription factors play a significant role in promoting species lipogenesis. And again, the activation of the insulin-like growth factor 1, insulin-like growth factor 1 receptor, phosphoinositide 3 kinase, ACT pathway results in the suppression of p53 activity by phosphorylation of the p53 binding protein mouse double minute 2 or MDM2. This phosphorylation enhances MDM2 capability to degrade p53, and by inhibiting p53, MDM2 supports cell survival and advances cell cycle progression. The other important effect of insulin-like growth factor 1 is the activation of mTOR-C1. Actually, elevated expression of mTOR and enhanced activation of mTOR-C1 have been observed in the epidermis and sebaceous glands of individuals with acne. The intake of Western diet leads to excessive stimulation of the insulin-like growth factor 1 ACT mTOR-C1 signaling pathway. And recent findings suggest that milk acts as a sophisticated endocrine signaling system in mammals, promoting mTOR-C1 dependent translation essential for postnatal growth. As we all know, acne has been linked to conditions related to insulin resistance, particularly polycystic ovary syndrome, which leads to hyperinsulinemia. Insulin and insulin-like growth factor 1, being closely related growth hormones, have the capability where the insulin can bind to and activate both insulin receptor and insulin-like growth factor 1 receptor. Both insulin and receptor and insulin-like growth factor 1 receptor share similar downstream signaling mechanisms involving the phosphorylation of a family of insulin receptors, substrates, and the activation of phosphoinositide 3 kinase ACT mTOR-C1 pathway. Milk is characterized by high insulinemic index, meaning it can significantly raise insulin levels, consuming large amounts of milk in contrast to meat has been shown to increase both serum insulin levels and insulin resistance. Therefore, milk intake enhances insulin and insulin-like growth factor 1 signaling, which is an important process for stimulating mTORC1 driving postnatal growth and anabolic activities. On the other side, metformin treatment is known to reduce insulin resistance, thereby diminishing insulin signaling towards sebaceous glands. Now I'm going to talk about hypoxia-inducible factor 1 alpha and leptin. In 2014, Damby proposed a theory connecting comedogenesis and inflammation in acne through ductal hypoxia. It is suggested that ductal hypoxia alongside mTORC1 activation and the resulting increase in hypoxia-inducible factor 1 alpha could increase the secretion of leptin from sebocytes into the rising sebum. This in turn might stimulate the proliferation of nearby infundibular keratinocytes, thereby encouraging comedogenesis. Hypoxia-inducible factor 1 alpha, the primary transcription factor involved in hypoxia, has its translation upregulated by the activated mTORC1. Hypoxia-inducible factor 1 alpha is known to transactivate the promoter of human leptin gene. Studies in human keratinocytes have demonstrated that leptin can mediate myotogenic stimuli, thereby promoting the proliferation of keratinocytes. Also, the increase in pro-inflammatory cytokines induced by leptin in normal human keratinocytes is primarily controlled through STAT3 signaling, a pathway that is also stimulated by the overactivation of mTORC1. Hypoxia-inducible factor 1 alpha also operates through the retinoic acid receptor-related orphan receptor gamma T, or ROR gamma T, to facilitate the differentiation of T helper 17 cells, so serving as a key metabolic reprogrammer in the inflammatory cells and promoting the expression of inflammatory genes. Also, hypoxia-inducible factor 1 alpha plays a significant role in maintaining the balance between T helper 17 cells and regulatory T cells. There is a lack of information regarding the impact of benzoyl peroxide, the most frequently used topical anti-acne medication, on the expression of ductal hypoxia-inducible factor 1 alpha in acne-affected skin. However, it has been reported that minocycline, the oral antibiotic often prescribed for treating inflammatory acne, leads to proteasomal degradation of hypoxia-inducible factor 1 alpha under hypoxic conditions. Also, azelaic acid might have effect on hypoxia-inducible factor 1 alpha, as it is recognized for inhibiting anaerobic glycolysis. It was found that activated p53 leads to a reduction in hypoxia-inducible factor 1 alpha protein levels through enhanced proteasome-dependent degradation. On the opposite side, the activation of ACT reduces the p53-mediated degradation of hypoxia-inducible factor 1 alpha. And a side note that increased expression of hypoxia-inducible factor 1 alpha have been recently observed in the lesional skin of patients with acne inversa or hidradenitis suppurativa. Now I'm going to talk about infundibular GATA binding protein 6. The transcription factor GATA binding protein 6 or GATA 6 regulates the proliferation and differentiation of keratinocytes, playing a key role in preventing the hyperkeratinization of the infundibulum. This process is a critical pathological event in acne vulgaris known as comedogenesis. Researchers have identified retinoic acid response elements in the potential regulatory region of GATA6 gene, highlighting retinoic acid as a vital signal for inducing the transcription of GATA6. The use of all trans-retinoic acid or ATRA for topical acne treatment, and the systemic administration of isotretinoin, a precursor of ATRA, may help to normalize the disturbed follicular keratinization and facilitate comedolysis by inducing the expression of GATA6. Significantly, the expression of GATA6 is also enhanced by the activity of FOXO1. GATA6 contains three potential binding sites for FOXO1. Inhibiting ACT leads to a decrease in phosphorylated FOXO1, so increasing the nuclear presence of FOXO1 and consequently boosting the expression of GATA6. On the other hand, heightened insulin-like growth factor 1 ACT signaling could reduce the expression of infundibular GATA6 and so promoting comedogenesis through the suppression of both nuclear FOXO1 and GATA6. Moreover, mTOR-C2 signaling, which is an androgen-driven pathway that activates ACT, also negatively regulates GATA6 expression in a FOXO1-dependent manner. Therefore, the enhancement of insulin-like growth factor 1 ACT signaling and androgen mTOR-C2 ACT signaling collectively contribute to the suppression of GATA6. Overexpression of GATA6 has been found to inhibit the expression of androgen receptor, suggesting that the increased infundibular androgen receptor signaling occurs in conditions where GATA6 levels are diminished. Another growth factor is transforming growth factor beta. Genomic and experimental data indicate that transforming growth factor beta signaling is diminished or reduced in the sebaceous glands of individuals with acne. The presence of transforming growth factor beta downregulates genes essential for sebaceous lipid production and sebocyte differentiation, like fatty acid desaturase 2 and BPAR gamma. This leads to a reduction in lipid accumulation via a pathway that is dependent on transforming growth factor beta receptor 2 and SMAD2. And there is a significant molecular interaction between GATA6 and transforming growth factor beta, where GATA6 serves as a key signal for the activation of transforming growth factor beta. Now I'm going to talk about the role of Propionibacterium acnes. Acne has been associated with disrupted innate immune response to Propionibacterium acnes, which leads to enhanced differentiation of T helper 17 cells and elevated levels of interleukin 17. This process contributes to the sebocyte-mediated shift of cutaneous T cells towards a T helper 17 phenotype. Studies have shown that membrane fractions from Propionibacterium acnes can elevate the expression of insulin-like growth factor 1 and insulin-like growth factor 1 receptor in the epidermis of explants. This increase potentially leads to a reduction in the expression of GATA6, which is mediated by FOXO1. Propionibacterium acnes also stimulates the expression of toll-like receptor 2 and toll-like receptor 4 on keratinocytes, a mechanism that could be vital in the inflammation associated with acne. This is because the activation of toll-like receptor 2 initiates an inflammatory transcriptional program in T helper 17 cells. Biofilms formed by Propionibacterium acnes, as opposed to its free-floating bacterial form, are marked by the upregulation of stress-induced genes and an increase in the expression of genes that code for potential virulence-associated cyclic AMP factors. So overall, the signaling derived from Propionibacterium acnes contributes to the range of transcriptomic abnormalities observed in Acne vulgaris. Now let's go and talk about the second half of this video, which is about transcriptomics in acne treated with isotretinoin. Unlike tretinoin, which is also known as all-trans retinoic acid or ATRA, Isotretinoin shows minimal or no affinity for binding to cellular retinol binding proteins or nuclear retinoic acid receptors or retinoid X receptors. Instead, isotretinoin may function as a prodrug undergoing intracellular conversion into metabolites that act as agonists for retinoic acid receptors and retinoid X receptors. First, I'm going to talk about sepocyte stem and progenitor cells. Recent research has focused on the role of stem and progenitor cells in regulating the development, physiology, pathology, and aging of sebaceous glands. Some researchers have reported that sebaceous glands primarily undergo self-renewal through dedicated stem cell pools during normal functioning. Retinoids are vital in the regulation of stem cell differentiation. The continuous upregulation of p53 and FOXO1 induced by isotretinoin may lead to the depletion of the progenitor cell pool, which in turn could result in the involution of sebaceous glands. As I have just mentioned, the second important effect of isotretinoin is the effect on p53, FOXO1, and FOXO3. Oral isotretinoin stands as the most effective therapy for resistant and severe acne, positively affecting all major factors involved in acne's pathogenesis. Experimental studies have validated that isotretinoin increases the expression of FOXO1 and FOXO3 proteins, indicating alterations in FOXO-mediated gene expression or the translocation of FOXO proteins. Experimental studies have demonstrated that treating primary human keratinocytes with isotretinoin results in an increased expression of the p53 protein. So in this diagram, we can see the main function of isotretinoin in acne is through enhancing the expression of FOXO1 and FOXO3 and increasing p53. So this will affect most of the pathogenesis of acne vulgaris. p53, FOXO1, and FOXO3 are basic negative regulators of cell cycle progression and cell proliferation. And as a pro-apoptotic transcription factors, they exert synergistic effects in promoting catabolism, autophagy, and apoptosis. p53 acts to inhibit the insulin-like growth factor 1 phosphoinositide 3 kinase act mTORC1 signaling pathway at various regulatory checkpoints. Collectively, the upregulation of p53 induced by isotretinoin effectively counterbalances the hyperactive insulin-like growth factor 1, insulin-like growth factor 1 receptor, phosphoinositide 3 kinase act mTORC1 signaling pathways and potentially the mTORC2 signaling pathway as well in patients with acne. Isotretinoin reduces the activity of both insulin-like growth factor 1, insulin-like growth factor 1 receptor, and androgen receptor signaling pathways, which are two key interesting routes involved in the ACT mTORC1 driving pathogenesis of acne. Let's talk about the third effect of isotretinoin treatment in acne, which is GATA6 upregulation. As I have just mentioned in the first half of this video, diminished expression of infundibular GATA6 has been associated with disturbed follicular keratinization leading to comedo formation in acne patients. Systemic isotretinoin treatment primarily affects the upregulation of infundibular GATA6 expression in two significant ways. In the first way, following the isomerization of isotretinoin into its active form, ATRA or all trans-retinoic acid, ATRA will activate retinoic acid receptors, which in turn enhance GATA6 expression by binding to retinoic acid response elements located on GATA6 promoters. The second way is through the upregulation of nuclear FOXO1 mediated by p53 that will activate the three FOXO1 binding sites on GATA6 promoter, thereby boosting its expression. Also, upregulated GATA6 acts to suppress the activation of androgen receptors, creating a synergistic interaction that helps to normalize the disturbed infundibular keratinization or comedogenesis observed in patients with acne. The fourth effect of isotretinoin is perilipin 2 mediated suppression of comedogenesis. The protein composition of lipid droplets has been recognized as a significant factor affecting various aspects of lipid droplets biology. Among these proteins, perilipins are the most extensively researched and are the most abundant proteins found on the surface of lipid droplets. Recent characterizations of lipid droplets protein expression in sebaceous glands have shed light on their importance in sepocyte physiology, sebaceous lipogenesis, and the development of acne. Perilipin 2, a major perilipin, plays a vital role in sepocyte differentiation and in regulating sebaceous lipid accumulation and the size of sebaceous glands. A key function of perilipins is to limit the access of lipases to lipid droplets, thereby inhibiting lipolysis, often referred to as gatekeeper effect on lipolysis. Some researchers recently noted a reduced expression of perilipin 2 in the infundibular regions of acne patients when compared to individuals without acne. Researchers have identified a possible role for p53 in the upregulation of perilipin 2 in sepocytes. The upregulation of p53 mediated by isotretinoin treatment may increase the expression of perilipin 2 in sepocytes, thereby inhibiting the hydrolysis of sebaceous triacylglycerol and reducing the release of comedogenic free fatty acids. The next effect of isotretinoin is on sepocyte autophagy and apoptosis. The upregulation of p53 induced by isotretinoin treatment is believed to be responsible for sepocyte apoptosis and the reduction of sebum production, which are key therapeutic effects in the treatment of acne. p53 is known to increase the expression of pro-apoptotic proteins such as tumor necrosis factor-related apoptosis-induced ligand, FOXO1 and FOXO3. On the opposite side, cervivin, a member of the inhibitors of apoptosis gene family and found to be overexpressed in serum of acne patients, is downregulated by p53. And it's well recognized that there is a significant interplay between autophagy and p53. Autophagy acts to suppress p53, while on the other hand, p53 serves to activate autophagy. Let's talk about the sixth effect of isotretinoin, which is actually a side effect, which is teratogenicity and neural crest cell apoptosis. Neural crest cells and cells derived from the neural crest, such as neuroblastoma cells, are highly sensitive to apoptosis induced by isotretinoin. Neural precursor cells have several p53-dependent apoptotic pathways. Evidence from translational studies suggests that the upregulation of p53 caused by isotretinoin treatment leads to apoptosis in the neural crest cells, which is proposed to be the primary mechanism underlying the teratogenic effects of isotretinoin. Another side effect of isotretinoin treatment in acne is depression and impaired hippocampal neurogenesis. The issue of whether isotretinoin leads to depression and anxiety in patients with acne remains a matter of debate. It's recognized that a subset of patients may be susceptible to developing depression or suicidal thoughts during isotretinoin therapy. The hippocampus is a key area in the brain where new neurons are continuously generated, a process known as hippocampal neurogenesis. In individuals with depression, reduced hippocampal volume and decreased the number of hippocampal neural progenitors have been observed. The upregulation of p53, FOXO1, and FOXO3 induced by isotretinoin treatment could potentially have a negative impact on hippocampal neurogenesis, potentially elevating the risk of depression in individuals who are predisposed to this condition. The next side effect of isotretinoin in acne is reduced ovarian reserve and granulosa cell apoptosis. Systemic isotretinoin treatment also affects the pituitary ovarian axis, leading to mild suppression of various pituitary hormone levels, including growth hormone, thyroid stimulating hormone, prolactin, adrenocorticotropic hormone, and luteinizing hormone. Actually, follicle stimulating hormone and luteinizing hormone are essential for the maturation of ovarian follicles. The impact of isotretinoin on the pituitary ovarian axis and the granulosa cells could be attributed to the overexpression of p53, FOXO1, and FOXO3 signaling. This overexpression may lead to the suppression of gonadotrophins and apoptosis of granulosa cells. Another important side effect of isotretinoin treatment in acne is the hypertriglyceridemia. The increase in p53 and FOXO1 levels caused by isotretinoin treatment offers a logical explanation for the hyperlipidemia often observed in individuals taking isotretinoin. This hyperlipidemia is primarily due to the liver's overproduction of very low density lipoproteins that contain apolipoprotein P100. The production of apolipoprotein B or B100 in the liver is essential for the formation and release of very low density lipoproteins. The ApoB gene, which is vital in this process, has been identified as a target gene for p53. The process of loading hepatic triglycerides onto ApoB100 is carried out by the microsomal triglyceride transfer protein, which is called MTTP, which is vital in the assembly and secretion of very low density lipoproteins and is stimulated by FOXO1. Also, FOXO1 activates the expression of apolipoprotein C3 in the liver. Research has demonstrated that ApoC3 significantly prevents the hepatic uptake of very low density lipoproteins and intermediate density lipoproteins, as well as inhibits the activity of lipoprotein lipase. Let's talk about another side effect of isotretinoin treatment in acne, which is dry skin. Dry skin is the most frequently observed mucocutaneous side effect of oral isotretinoin treatment. The increased transepidermal water loss that occurs as a result of oral isotretinoin treatment has been linked to the upregulated expression of aquaporin 3. Aquaporin 3 regulates a channel responsible for transporting water, glycerol, and hydrogen peroxide, playing a vital role in several processes related to keratinocyte function. Aquaporin 3 is identified as another target gene of p53. Intracranial hypertension is another side effect of isotretinoin treatment in acne. Intracranial hypertension, also known as pseudotumor cerebri, is a potential side effect of oral isotretinoin treatment. Aquaporin 1 is vital in facilitating the movement of water through the epithelial cells of the choroid plexus. Aquaporin 1 is extensively present in the human brain and is involved in the secretion of water into the subarachnoid space. It has been proposed to play a significant role in both idiopathic and drug-induced intracranial hypertension. The overexpression of p53 in choroid plexus epithelial cells induced by isotretinoin could account for the development of intracranial hypertension in individuals who are predisposed to this condition. The inflammatory flare with the initiation of isotretinoin is another well-known side effect. And in a trial to understand its mechanism, interleukin 1 beta messenger RNA and its active processed form are highly present in inflammatory acne lesions. Caspase 1, also referred to as the interleukin 1 converting enzyme, is a protease that processes the key pro-inflammatory cytokine interleukin 1 beta. It converts the inactive precursor pro-interleukin 1 beta into the active secreted form of interleukin 1 beta. Besides its role in interleukin 1 beta processing, caspase 1 also plays a vital role and conserved role as a protease involved in cell death. And it was found that CASP1 is a target gene of p53. Therefore, the initiation of isotretinoin therapy, which leads to the upregulation of p53, might explain the initial inflammatory acne flare at the beginning of isotretinoin treatment, as well as the occurrence of acne fulminans after starting isotretinoin therapy. There is increasing evidence indicating that various signaling molecules known to regulate programmed cell death also have the ability to modulate inflammasome activation. As a conclusion, acne vulgaris, a widespread inflammatory skin condition prevalent in Western countries, is associated with both genetic and various environmental factors. A Western-style diet characterized by high consumption of hyperglycemic carbohydrates, milk, and dairy products is particularly linked to the development of this condition. It's important to recognize that the transcriptomic alterations identified in the context of acne vulgaris show considerable similarity to the transcriptomic patterns observed in cancers that are common in Western civilizations, particularly prostate and breast cancer. Given these findings, it's not surprising that epidemiological studies have found a link between severe acne during adolescence and an increased risk of developing prostate cancer and breast cancer later in life. In contrast, the transcriptional changes brought about by isotretinoin treatment have been found to be beneficial in the prevention and treatment of various types of cancer. Lastly, I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell, and thank you.